So good morning, everyone, and welcome to this ISMB 2020 conference on the high throughput sequencing track. And my name is Mingze Gao from the University of Hong Kong. And today I'm very glad to bring you the latest research of my lab called UniTVelo, temporary unified RNA velocity reinforced single-cell trajectory inference. So basically, RNA velocity is a very new concept introduced in 2018, which can be used to retrieve dynamic information for cellular trajectory and predict its future states. And in 2020, and by Bergen's paper, this method has been extended to ISMB package with the covariance-based stochastic mode and likelihood-based dynamic mode. However, in many different biological systems or scenarios, we found that the velocity estimation are quite often to return the erroneous results, that the performance are compromised. We suspect that this partly due to the model requires genes to have a strong dynamical trace or lack of temporal regularization. So we propose UniTVelo, a statistical RNA velocity analysis framework via spliced RNA-oriented function. Okay, so we have two innovations or two advantages over the previous framework. The one is using RBF kernel to model transcriptional dynamics. So if you could look into the panel A here, in the methodology part, we have patterns of induction, repression, and transient dynamics of each gene, which can be modeled by a single RBF kernel by adjusting the parameters with different ranges. And for instance, the tau G part. So for each different tau G value here, we actually we could model the different expression profiles of different genes. And the second innovation of our kernel, of our model, is the supports unified waiting time across genes for cells when estimating the velocity-based related parameters. We've used this kind of method because that we found that most of genes expression profiles, we don't have a very strong dynamical trace. And most of they are just constantly increasing or constantly decreasing along the differentiation path. And sometimes they have the multiple rate kinase genes. For instance, the genes have a transcriptional boosting at a later stage, which are quite contradictory with the original hypothesis of the ISMB package. So we've introduced this unified time mode. So basically, for the more parameter inference part, we have two sets of parameters. One is gene-specific parameter theta, which define the phase portraits of each gene. And we have cell-specific time points t, which are assigned by minimizing the Euclidean distance to the phase portrait trajectory. We could infer this kind of trajectory for each different genes. So we have a unified time mode, which enables the discrepancy between genes directionality to be minimized. And we also retain an independent mode here to resolve some very complex data sets if the unified time mode failed to capture the correct directionality. For instance, the data sets with cell circles or data sets that are with very sparse clusters, or they have multiple, like 4 to 5 different branching sites, that they are complex enough and not to use unified time mode to model. But after testing in more than 10 different real data sets, we found that for most of the scenarios that our unified time mode is quite sufficient to get a correct and satisfied RNA velocity result. So for the results part, first, if we look into this panel A and B here, which is the mouse eraser hematopoiesis data sets, we know we have 5 different clusters here, from blood progenitor cells to the mature eraser cells. And the correct or the biological ground truth direction is from progenitor cells to eraseries. And from the results here, we know that ISMB actually captures a reversed or distorted velocity field, whilst our UniTVelo could give you the correct directionality from immature to mature cells. And if we look into the specific genes, and here we take a Merck gene as example, we know that these kind of Merck genes have a transcriptional boosting at a later stage of the eraserate, which is the main reason caused why ISMB failed to work on this kind of data sets. So for this merged gene from the face portraits, we know that HiTSeq actually gives us a reverse direction and mark this gene as a repression gene. However, in our UniTVelo package, we've successfully identified this gene is actually an induction gene, that the expression profiles is actually increasing as the differentiation goes along with the time. So our second example is the human bone marrow development data set. We have one starting size, the stem cells, and 3 terminal sites, CLP cells, monocytes, and erythroid. So we have also successfully identified these 3 branching scenarios, whilst as well actually have our reversed direction from monocytes or erythroid to stem cells, which is quite contradictory with our biological ground truth. And we also support the using different evaluation metrics such as the model regression R-square to identify the informative genes during the differentiation process or identify distinctive branches. So from some example here, we know that the higher R-square values compared with lower R-square values could better help us to identify this kind of informative genes. So this is the main idea of our paper, and please check out the details and other different results. If you're interested in this paper, you could scan this QR code listed here, which could direct you to the bioRxiv site, which we published a few months ago. And thanks for listening, and I'm happy to take any questions you may have.